All right, so I know these are really specific papers. I'm going to talk to you guys about different visualization techniques to reveal ecological information from soundscapes. So really directed towards my dissertation research, looking at soundscapes and marine systems, though these are terrestrial examples. Oh, okay. All right. So if we look at this picture of a meadow and forest, and if you're standing in the middle of this field, what might you hear? I think I would hear things like birds, wind, insects, and maybe planes flying overhead. All of these different sounds and others form what's known as a soundscape. And so this is the collection of sounds of a different habitat or environment. And more recently, a lot of researchers have been looking at using soundscapes to monitor different habitats that might be more difficult to monitor using field-based techniques. And so the benefits of acoustic monitoring are, there's a lot of them, but I'll go over a few of them. The first is temporal and spatial cover. So with traditional field-based sampling, you have to go out into the field, sample the environment, and then bring it back to the lab to analyze it. And so this provides snapshots of how the habitat is performing. With acoustic monitoring, these devices can be put out and record continuously for days, weeks, months, or even years. In addition, you have higher spatial cover, so instead of having to go to multiple different sites, you can put these out at different sites. They'll record simultaneously, and so you're getting an idea of how all of these different locations are changing over time together. Second is persistence. This data can be collected and then saved forever. Though my advisor likes to save all of his field samples, they get really icky over time, and so you're losing that information. You can't go back and look at your samples later on. The sound files can get saved. They can be analyzed later on when new tools come about to better analyze them. And then lastly, it's minimally invasive, and I think this is the key here. These devices are just strapped to a tree or a fence post and left out there. So you're not consistently going back, physically sampling the environment. They're just passively recording. In addition, you're not interacting with the environment or the animals themselves, and so you're not directly influencing animal behavior. So you're just recording what's happening naturally in those habitats. So despite these benefits, there are some disadvantages, and that mainly comes with the analysis of these long-term audio recordings. They're really large volumes of data, and they can happen over extensive timescales. You also have your different sites that you're recording. And so the soundscape provides all this information because it's created by and influences behavior of different animals, but we don't really know how to get this information out of these audio files. And so where a lot of the research is going in soundscape ecology is how do you distinguish signal from noise, and how can you extract ecologically relevant information from these files to answer your questions that you're trying to answer the questions that you're posing about these environments. So right now, the general approach to analyzing this data are three different ways. I'm sure there's more, but these are the main ones. So general acoustic characterization. So this is just looking at how loud sound is and what frequencies are present. More recently, more sophisticated techniques have come around, and that's automated species recognizers. So building a detector to detect a certain species in the sound file, and so using that detector to run across all of your recordings and saying when that species is vocalizing. And more recently, there's machine learning techniques, so training a model to identify certain sound signatures that you're able to identify in recordings, and having the computer do that for you. There are challenges with all these. The first being that, especially with the first method, is that there's no real ecological information. You're just getting information on how loud sounds are over time, but not which species are making those sounds and how they're interacting with the environment. And then the more sophisticated techniques, though, they provide some information about the species that are present. They're difficult and time-consuming to code up, and are often specific to single species that you know are there, and won't tell you anything about other species that are present that you might not know about. And so when approached with this problem and looking at the different papers, I wanted to look at visualization tools that could help filter down my data and look at different tools to identify the ecological patterns in audio recordings. And so the first paper looked at long-duration false color spectrograms. The second used clustering techniques and different visualizations to look at long-term audio data. And the last is a complete interactive visualization system to look at soundscape data. So I still have some more introduction material because I feel like all these papers were based on similar concepts. And so that was using spectrograms and then calculating different indexes on those spectrograms. And so what is a spectrogram? So a spectrogram is a visual representation of sound. Shown here, it shows time on the X, frequency or pitch on the Y, and then sound intensity and color. So cooler colors in blue range represent quiet sounds, and brighter red colors represent louder sounds. And so you can get an idea of how loud certain sounds are over time in these recordings. Now to calculate the different indices that they use in the papers, they split up these continuous recordings into different data frames, mostly of 1 minute duration. So you take your spectrogram or your audio file, split it into 60-second windows, and then once you have your data frame, they calculated those indices or metrics, and they're kind of interchangeable. And these metrics summarize some aspect of the distribution of the acoustic energy in a recording. And so they did that for the entire recording, but then also broke it up by different frequency bands, because different species will vocalize at different frequencies. So some might have really high-pitched vocalizations, others might have low. And so getting an idea of how the energy changes over time and frequency is important to characterizing the soundscape. And with that, we'll get on to the first paper. So long-duration false color spectrograms for detecting species in large audio data sets. And so the goal of this paper was to take these audio recordings and then make a figure that you can easily pick out important features in the recordings. So to do this, they calculated the different acoustic indices from the original sound recording, and then selected indices that were independent and then best described the features of interest that they were interested in. So if they were interested in bird calls, what index best summarized bird calling. And then they plot each index to the red, green, and blue channel of a colorized spectrogram. And so I'll build one for you now. So here I calculated ACI from a recording, and so that's really good for identifying biological sounds in general. Then they calculated spectral entropy, which is good for detecting strong but infrequent calls. And then lastly, they calculated cover. And so this is how loud sounds were in different bins of the spectrogram. And this is really good at picking out continuous choruses. So then they plotted this to the red, green, and blue and come up with this false color spectrogram here. And so with the false color spectrogram, you can easily pick out different areas of interest that you might want to go into. So like this green area here is highlighted with the spectral entropy, which again was a strong but infrequent chorus. And so going into the original recording, you might identify what that species is, and now easily be able to track it through time in these spectrograms. So the paper that you all read looked at different species and how these spectrograms can be used to track them over time. And so I'll just go through one here. On the right, you have the original grayscaled spectrogram. So this is just the original recording. Darker colors indicate louder sounds. And so in that 5 seconds of recording, you can see the call of this burrowing tree frog. And so in the 5 seconds, there's 3 calls. Now in the false color spectrogram, this is a longer time scale, so this is showing 3 hours. You can see that this call is being lit up by the feature that's plotted in green. And so you can see how long that species is chorusing for, the start and end time of that chorus, all based on this spectrogram. In addition, they also point out this feature at around 5,000 hertz, which is an insect chorus, I believe. And again, it's really highlighted in this bright pink color and easy to pick out, versus in the original spectrogram, it might not be as easy to pick out. They also showed these ribbon plots, which I thought were really interesting. And so you can kind of see the first one here. This is 24 hours of recording on March 4th in 2016. And so when we plot all of them on the same page, I think this is 21 days of recordings, you start to notice some really interesting patterns that emerge over time. So I'll point out some of these now. So this green feature starts at around 6 a.m. and goes throughout most of the day. And so this is typical of bird chorusing. So you can see when these bird choruses start and end throughout a day. And then most interesting for them was this bat call detection. So bat echolocation calls can be detected at night in this hot pink color. And so you can see differences in timing between different species, when they start vocalizing, when they end vocalizing, how it progresses over a daily scale and also seasonal scales. The next paper was revealing the ecological context of long-duration audio recordings of the environment through clustering and visualization. And so one of the big motivations for their work here was reducing the size of their data set so they can more easily see patterns in the different species of interest. So we'll go back to our data processing and reduction method here. So again, here's a spectrogram. They had continuous recordings that they broke up into 1-minute segments and then calculated 12 acoustic indices on those segments. And that resulted in a feature vector for each minute of recording for a total data set which was the size of the number of minutes of recording in the 398 days times those 12 acoustic indices. They then used that feature vector to cluster, and so they had this hybrid method where they did k-means clustering and then hierarchical clustering to try to pull out the different groups of animals of interest. And so they optimized their cluster size by assuming that similar biological sounds in 2 recordings would be more similar the closer they are in space and time. So if they're from the same site and have the same types of animals, those recordings would cluster closer together. And then acoustic signatures of 2 recordings with similar biological sounds would be closer than 2 with dissimilar biological sounds. So the results, they ended up with 60 clusters. 42 of those were dominated by a single sound source, 11 by 2 or more sources, and 7 were difficult to label. So that was pretty good that they were able to identify what these clusters shook out to be. They identified 7 major sound sources within these clusters, and that included rain, birds, cicadas, wind, planes, quiet, and insects. And so, most notably, they took their really large data set.
Sometimes just turning it on and turning it off.
To 1.1 million features, which are classified into 7 different groups.
Apologies for the technology.
Oh, well, now we're getting to the visualization part.
Make some sounds.
Okay, so once they had those group clusters, I'm just going to keep talking, they visualized them in different ways to start to get at how these different sounds were appearing over time and space and interacting with each other. And so the first plot I'm going to show here are the histograms. And so this is showing the average number of minutes per month in each 2-hour period. So the bars correspond to 2 hours and how many minutes during those 2 hours that cluster was present, or that cluster was classified as. And then each little brick is the month that it was. And so if we're looking at Orthoptera, which is just insects like grasshoppers and crickets, you can see that they're most dominant in the summer. So this is in the southern hemisphere, so December or summer. And that it happens mostly at night. So their peak in vocalizations are at night. And then if we add different species, so birds and cicadas, you see birds vocalize earlier.
Close that message. I gotta get those notifications.
Birds start calling earlier in the season, so in spring, and are most dominant in midday recordings. And then cicadas are more infrequent, and they emerge in late summer, mostly during the daylight hours. So you can see, starting to see different temporal patterns in the different species and perhaps how they might interact together over time or with their environment. I thought this plot was really interesting. So this is their diel plot, and this is showing basically all of their data in one plot. And so every pixel is that 1 minute recording classified as one of the 7 different sources. And so you have time of day on the x-axis, and then each of the recordings minutes on the each of the recording day times or days on the y. And so we start to see really interesting patterns here. The green, again, is birds, and so you can see their morning chorus, how it progresses through a year. It goes right in line with when daylight hours get longer and shorter. So in summer, when daylight is longest, you see that they start chorusing earlier and end later in the day, versus in the winter months, like up here, they're chorusing later in the morning and ending earlier in the day. If we move on to nighttime, you see that cicadas are really prevalent at nighttime, or no, this is insects. Insects are prevalent at nighttime, and again, having that seasonal pattern, you don't see them as much in winter and are really prevalent in summer. The last visualization I'll go over from this paper are the polar histograms, and I've been seeing a lot of these in soundscape papers that have been coming out because they're a really great way to see all of your data at once. And so this is showing the proportion of minutes per day in each of the classes that they identified. And it's really illustrating broad scale changes in the soundscape over time. And so we're starting with their first recording month in June, going all the way through to July. So you see when birds start to vocalize throughout the year, they get higher and then get lower again, so that's the green. And the same thing with cicadas emerging at a certain time. And then most interestingly is they had this interaction between rainy files and insect files. And so if you start to see these dark blue bars, they're almost always followed by a couple of days of really high insect activity. And so again, you're starting to get some more information from these files all in one figure. The last paper is a visualization framework for feature investigation in soundscape recordings. And so this was an entire system of taking your data and then visualizing it and being able to listen to files and seeing the different features and how different files were classified using the feature extraction. So their objective was to develop a user-driven tool to assist in analyzing and investigating large sets of environmental recordings. And so essentially how they did this is they used audio feature extraction, and then the system would be able to identify relevant acoustic events using both visualization and the associated audio file, and then using what you identified in step 2 to further identify events that are similar and then characterize the soundscape overall. And so the main visualization here was the RADVIS, which I had never heard about before. And so the RADVIS works by having anchor points along the outside of the circle. And so those are the features of interest. You can see all these here. And then the individual files were plotted as points. And so a point that is most influenced by a certain feature is pulled towards that part of the anchor. So these green points are most influenced by these features down here, whereas these orange points were most influenced by these features over here. And so the best way to look at this is to look at it. Oh no, did it get closed?
The Internet Explorer.
And Internet Explorer, yeah.
Oh, okay. So they had databases already loaded in here, which is really cool. So if you wanted to go and just explore soundscapes of Costa Rica, you could do that. So it's pretty fast too. So this database, Costa Rica 2, was loaded and plotted the different features here that were most important based on the number of clusters entered here. And so if you look at the correlation matrix, which they talked about a lot, you can see which features were included in those clusters. And so the anchor point around the RADVIS here was the most important feature in the cluster shown in this correlation matrix. And so all the blue cluster features are represented by spec mean on the Rad-Viz plot there. What also I think is really cool is if you click on a certain point, the spectrogram comes up, and you're able to play that recording and listen to what's there. Pretty peaceful. So this is a relatively noisy recording, and then you're able to look at how different features are summarized by the different features in the spectrogram. And then you can add labels saying, like, okay, this is a certain insect, and then it'll cluster out all the files that are similar in terms of the features that you've highlighted in this recording here. And just to show that these are different from features up here, we can look at another recording. This one's much quieter, and so it's summarizing some different type of information. If we play it, you don't really hear much. So I didn't get a chance to like really look at this. This is pretty cool though, that you're able to see the data and then hear the data, annotate it, and kind of do this iterative process of where you're looking at the different features, which features are best summarizing that vocalization that you're interested in, and then being able to redraw this plot and see if they break out better than the original one. All right, so in summary, there's increasing amounts of acoustic data, and so there's this need for tools to extract and synthesize ecological information from these data sets. And then the visualization tools that I discussed today are able to convert these large amounts of data to interpretable images that were able to start answering ecologically relevant questions from the information gained from these recordings. And with that, I'll take any questions.
Has it been applied to fish yet?
No. So I was going to load some of my files to that visualization framework. There's a lot of different settings you kind of have to know about before loading it in, and there's differences between terrestrial soundscape recordings and underwater soundscape recordings. So I didn't really have the time to go through that, but I think it'd be a really neat tool for just initial feature investigation, and I'd be interested to see how some of the recordings I have kind of cluster out using that method.
This is fascinating. Are people doing cityscape sounds as well looking at it this way as well?
I'd assume so. I'm really more involved in like the marine literature and then kind of advanced techniques in terrestrial kind of natural soundscape stuff. So I haven't looked a lot into like city soundscapes, but I'm assuming all of these can kind of be applied. I mean, it's kind of just scene classification, so that's kind of where the field is going. So this small section of recording, what is it kind of thing, and being able to automate that as much as possible, because you can't ever listen to all this data or even visualize all the data.
We have other questions? Otherwise, we're going to go. Let's go over the quiz. So, our two speakers are to. Can you lead the answers to